Hello, Bio30, Mr. Walker here. This lesson is going to be on fertilization and early embryonic development. We're taking a look at this first slide, which is a cut through the female's reproductive system. So some of the structures that we can hopefully identify here, the ovaries, or at least one of the ovaries. We can see the fallopian tube or the oviduct, the ovarian end or opening of the fallopian tube where we have the fimbriae. We have here now the exit into the uterus, and it looks like what they're showing us here is the entire uterus. So the cervix would be down in this region here, and down below that would be the vagina. And we can also see in the uterus, it also identifies here the inner lining or the endometrial lining of the uterus, as opposed to the thick muscular layer of the uterus itself. So those are the structures that we're going to be talking about with fertilization, implantation, and early embryonic development. So we'll start with the ovary and what's going on in the ovary. So we've already gone through ovarian changes that are taking place throughout the female's ovarian cycle or reproductive cycle. So where we're going to pick up with this one here is about halfway through the female's ovarian cycle, and that is the time that ovulation is going to be taking place. If ovulation is taking place and everything works as it should, these fimbriae should be drawing the egg into the fallopian tube. Cilia that are lining the fallopian tube will then start to move that egg in the direction of the uterus. If intercourse is taking place at the same time, there will be sperm that are deposited in the vagina. Those sperm will then need to swim up through the cervix and make their way into the uterus. So during sexual intercourse, it might be an enormous number of sperm that do end up in the vagina, somewhere on the order of perhaps 500 million or 500 million sperm in the vagina. They then need to make their way through the cervix, but quite often, at least when the female is not around the time that she is ovulating, the cervix is actually, for the most part, not penetrable by the sperm. The sperm cannot gain entrance for the most part into the uterus because there is what is called a mucus plug. If the female is ovulating, then around that time, the texture, the consistency of the mucus plug is going to change, and that's then going to allow at least some of the sperm to travel up through channels and into the uterus. So this will take a period of time, and by the time the sperm do reach the uterus, most of them actually will have not made it for multiple different reasons. Maybe they're not getting access to the fructose in the semen. Maybe they're just not good swimmers. Maybe they don't have a tail. Maybe they have two tails. Multiple reasons, but by the time the sperm reach the uterus, well, it might be down to about 500,000 out of the original 500 million that were again deposited in the vagina. So now the sperm are going to be traveling up, and contractions of the uterus are going to help the sperm to travel up through the uterus and toward the fallopian tube. So when they do reach the fallopian tube, now down to somewhere around, they say, about 6,000 sperm that are now left. So because there are two different fallopian tubes, what that kind of means is that, well, half of them will turn in the wrong direction, the other half will turn in the right direction, keeping in mind that in humans, it is not one egg that is released from each ovary, it is one egg between the two ovaries. In reality, probably this egg that is being released from the ovary when it is in the fallopian tube, it's probably releasing some chemical signals. So it's probably not out of the 6,000 that 3,000 are making the wrong turn. Probably most of them are heading up and swimming in the right direction, and that is going toward the egg that was released from the ovary, was drawn into the fallopian tube, and the normal site of fertilization is typically what is described as the upper 2/3 of the fallopian tube, and it is shown taking place right here, the process of fertilization. So when this contact is made between the sperm and the egg, it's not just one sperm, but it would be multiple sperm that are surrounding the egg, all of them releasing the contents from the acrosome. Remember that the acrosome contains digestive enzymes that are going to dissolve away the outer portion of the egg. Ultimately, though, it is just one sperm that will fertilize the egg. And when that happens, it's going to change the outer membrane of the now fertilized egg, and that's going to prevent any other sperm from gaining entrance into the egg. And again, where this fertilization does take place, at least where it should take place, where it normally takes place, is in the fallopian tube. So now what we have done is we have taken a haploid sperm. Remember that haploid means that it has half the normal number of chromosomes. We have an egg or oocyte that has half the normal number of chromosomes. And in the process of fertilization, where the sperm and the egg are meeting, now what we have are these two nuclei that are coming together. And that's going to generate the first cell of this individual that has two copies of each chromosome, in other words, a diploid cell. This is the fertilized egg, but the name that we will now call it is the zygote. Zygote is the fertilized egg. So it is from this original zygote or fertilized egg that all of the trillions of cells that eventually form will arise from. After fertilization, the cilia that are lining the fallopian tube are going to continue to now move the zygote in the direction of the uterus, but things are going to start to happen fairly quickly in terms of cell divisions. So very early on, just a few hours after this fertilization, this first cell, the zygote, is going to divide. So it's going to divide into 2 cells and then into 4 cells. And initially, it's just kind of a doubling like this, 8 and 16, 32, and so on and so on. All of this taking place as what I'll call the early embryo is now traveling through the fallopian tube from the upper 2/3 where fertilization took place and toward the uterus. So all of these divisions, they are mitotic divisions. And of course, mitosis is the process that is going to produce genetically identical cells. So all of these cells that we now see here, these ones at the 4-cell stage, all of these ones that are at about a 16 to 32 cell stage, a solid ball of cells that is now referred to as the morula. These are all genetically identical cells, and these arise through the process of what are referred to as cleavage mitotic divisions. Mitotic divisions generating, again, genetically identical cells. Sometimes what can happen is with humans, what if it's not just one single egg that is released? What if it's 2 eggs or 3 eggs, or what if it's 1 egg from each one of the ovaries? Well, if both of them become fertilized, remember that each egg is genetically unique. Each sperm is genetically unique. So if we do have the fertilization of more than 1 egg, that will result in twins. But in this case, they are referred to as non-identical twins. Non-identical twins. We can also have identical twins that form. So if we go back up to, say, this 4-cell stage, sometimes what happens is, well, these cells should stick together, but sometimes they don't. So perhaps we could have something like this, where these two become separated from these two, they pull apart, and then they begin to divide independently of each other. As they divide independently, eventually they will go through all of the normal stages of development as they would if they remained stuck together. And this would result in then identical twins because they did arise from the original single cell, the fertilized egg, that zygote in the first place. So that would be the source of identical twins. So eventually, about 4 days after fertilization, this is going to be our reference point now in terms of timing, fertilization. So about 4 days after fertilization, now what we have is a structure that's going to enter into the uterus, and this is referred to as the blastocyst. This one here, the early blastocyst. So whereas the morula was a solid ball of cells, The blastocyst is a fluid-filled ball of cells where we have some cells that are around the outside. We have some fluid, which is in the middle, what it has labeled as the blastocyst cavity. And we also have another cluster of cells at one end of the blastocyst, and this one is referred to as the inner cell mass. I'll just put it for now as the ICM, the inner cell mass. So again, this is the structure that we now see that pops into the uterus, and that is about 4 days after fertilization. So now this blastocyst needs to attach to the endometrial lining of the woman's uterus, and this might take a couple of days. There are molecules on the endometrium that need to match up with molecules in the outer portion of the blastocyst. And the outer portion of the blastocyst, it has labeled in this diagram here, it is referred to as the trophoblast. Oh, and we do have the inner cell mass, in fact. Let's label down here as well. So the trophoblast is going to make contact with the endometrial lining. And once we have this contact, not only is it going to touch, but it's going to start to grow into the endometrium itself. And this is now the beginning of what we do call implantation. And this process of implantation takes about 6 to 7 days. And again, our reference point is fertilization. So implantation is initiated about 6 to 7 days after fertilization has taken place. Now that's what should happen, but sometimes it doesn't quite turn out that way. So the egg, of course, may not become fertilized for multiple different reasons. Sometimes it may become fertilized, but then it doesn't make its way to where it should be, which is in the uterus. And typically the best location is the upper back portion of the uterus for implantation to take place. Remember that fertilization does normally occur inside of the fallopian tube right about here, and then it's undergoing all of these cleavage mitotic divisions. It's moving in the direction of the uterus, and it should make its way to the uterus, but sometimes, unfortunately, it doesn't, again, for multiple reasons. So it can get stuck in the fallopian tube, and if it does stick to the fallopian tube and try to draw nutrients out of the fallopian tube, like it typically would out of the uterus, there is simply not enough space for that embryo to grow. That is not what the fallopian tubes are designed for. So if this does happen, it's a significant risk to the mother that the fallopian tubes are going to rupture. This is going to result in severe bleeding or hemorrhaging, and that could result in not only, well, the death of the embryo, the early baby, but it can also result in harm to and possibly death of the mother as well. So this kind of pregnancy is what is referred to as a tubal pregnancy. A tubal pregnancy is one form of what is referred to as an ectopic pregnancy. An ectopic pregnancy is where there is implantation somewhere other than where it should be, which is inside of the uterus. So tubal pregnancies are one type of ectopic pregnancy, and they are the most common, but there are some other locations as well. Sometimes that egg may not be drawn into the fallopian tube at all, and it may end up floating around somewhere in the pelvic and abdominal cavity. So if this egg is not drawn up here, but ends up down here somewhere, those sperm that are swimming up through the vagina, the cervix, the uterus, well, they're going to keep swimming until they find that egg. And if they find it here somewhere, they're going to fertilize it, and then that embryo is going to look for a place to implant, which could be various different locations. This is not inside, this is outside of the ovary. It could be the entrance into the fallopian tube. It could be somewhere in the abdominal cavity. It could be on the outside of the uterus. So in all of these cases, this creates a little bit of a problem because there may actually be some space for that baby to grow inside of the abdominal cavity. But unfortunately now there is no way for that baby to get out. It cannot make its way out through the normal birth canal, which is passing through the cervix and through the vagina. So all of these are referred to as ectopic or ectopic pregnancies. There's one other one anyway that they do show on this picture here as well, and that is in the uterus, but it's right at the bottom, right at the cervix. So this is called a cervical ectopic pregnancy, and this can create some problems as well during the delivery process. We will talk later about the placenta, and that the placenta is the connection between the baby and the mother. And if there is a cervical ectopic pregnancy, that could result in the placenta coming out before the baby. The placenta usually comes out after, and that's why it's called the afterbirth. So that can present a little bit of a problem if that is the lifeline for the baby, and if it is delivered before the baby actually comes out. This slide here summarizes what I did already talk about in terms of the early embryonic development. All of this again is taking place inside of the fallopian tube. So we have the original egg once again, which has half the normal number of chromosomes. When we're talking about humans, that number is 23. The sperm that comes along, even though it is much, much, much smaller, it doesn't matter. It is also haploid, and it has exactly the same number of chromosomes, which is 23. So it's these two that we're adding together when we're forming the zygote. So we have the haploid number here, the haploid number here, 23 from one parent, 23 from another parent, and that gives us the diploid number, which is the 46 chromosomes, which we typically do have in all adult human cells. Then we have some cell divisions that are taking place. Again, these are mitotic divisions, and initially it's a doubling, so 1, 2, 4, 8, 16, and so on and so on. And you should know the name in addition to the zygote of some of these other structures as well. The morula is the solid ball of cells. The blastocyst is the fluid-filled ball of cells where we have the trophoblast on the outside and the inner cell mass on the inside. Already something else is taking place as well as we go through these different stages, early stages of embryonic development. These cells that are now part of the inner cell mass, they will actually form the baby. They will form the embryo, eventually the fetus and the baby when it is delivered. The cells around the outside, remember those are the trophoblastic cells, they form what are referred to as extraembryonic structures, and that includes structures that are not part of the embryo that are vital but are vital for the survival of the embryo, and that includes one that I mentioned like the placenta, the umbilical cord, other ones that we'll come across like the yolk sac and the amnion as well. But the inner cell mass, there are already some changes that are taking place here. So when we have the inner cell mass, maybe it's 100 or so cells, but at this point, they're all genetically identical, but they're already beginning to look and behave a little bit different because a process referred to as differentiation has already begun. Differentiation is the process whereby cells acquire their distinctive structures and functions. In other words, they become specialized. And already at the stage of the blastocyst, some of these cells have started to go down a certain route. Their fate is partially determined. So some of them will give rise to muscle cells. Some of them will give rise to liver cells, and eventually all of the other cells in the body. So this process of differentiation or specialization is already beginning, in fact, already at the stage of the morula, and probably even before that, it probably begins somewhere around the 8-cell stage. And after this, those cells, so going on from the 8-cell stage, not at the 8-cell stage, but after the 8-cell stage, differentiation would have taken place, at least partially. And it will continue on for weeks after that. What is then going to take place after implantation is a process that is called gastrulation, and this is where we do have, especially kind of focusing on the inner cell mass, the rearrangement of the cells. And initially from the inner cell mass, they're going to form into two different cell layers. So this cell layer that it shows right here, that is called the ectoderm. This one that it shows right here, this one is the endoderm, so they're initially adjacent to another or to each other. And then eventually there's a third one that's going to form. This one is going to be in the middle, and this one is going to be the mesoderm. So these three different what are called germ layers are formed through this process of gastrulation. So these three different germ layers again, this one here that they typically show in yellow, but that one there would be the endoderm. This one that I'm showing in blue, and they do typically use blue for this one. That would be the ectoderm, the ectoderm. And the third one that is sandwiched in between them, that one that they do show in red, that one is the mesoderm. So these will give rise eventually to different cell types, like the mesoderm will give rise to things like the heart and the muscle and bones. Endoderm, the lining of internal organs and fat. Ectoderm, things like the skin and nervous system tissue. Also, a process called neurulation does take place, where we do have portions of the brain and spinal cord that form. And again, these do form, nervous system tissue does form from that blue layer that we just saw, which is the ectoderm. So from the ectoderm, we're going to see this folding that's taking place. And through this folding process, it's going to form the neural tube. And it's inside of this neural tube that the brain and the spinal cord will eventually form. So I'm going to use this slide here to summarize pretty much everything that we've talked about so far. So we have our haploid sperm and egg. I'll just use the symbols N for these ones that are coming together. We form the zygote, which is 2N, and that process, you need to know about processes, that is the process of fertilization. Fertilization. Then what we have taking place over, well, a few of these. Here we do have mitotic divisions, and I mentioned this word as well, cleavage divisions. These are cleavage mitotic divisions. We also have the beginning of the process of differentiation also taking place in here. Cells begin to acquire their distinctive structures and functions. They become specialized. That is the differentiation. Once we do have the blastocyst, and now we've reached the stage of the uterus, so we're going to have implantation taking place. And once again, there is a time frame for all of this. So all of this from fertilization through to implantation, this is taking about 6 to 7 days. About 1 week after fertilization, implantation is going to take place. We again see a picture here of the blastocyst. So remember that in the blastocyst, we do have the trophoblast around the outside, and we have the inner cell mass on the inside, and we also find the fluid-filled cavity, the blastocele on the inside. So I'll just highlight that again here. This is the trophoblast. It gives rise to structures that we'll talk about later. I mentioned this, the extraembryonic structures, and just 3 of them that I've listed here, the chorion, the placenta, and the umbilical cord that we will talk about later. The rest, the inner cell mass, that's this part right here. That again is what is going to eventually form the embryo. So even though differentiation is taking place and has been taking place from about the 8-cell stage on, definitely more differentiation taking place here. We have a structure called the gastrula that is forming. That gastrula is what it makes up or is made up of the three different germ layers that we see right here. And those again are the ecto, the meso, and the endoderm. So this one here would arise through that process that we saw that is called gastrulation.